Welcome back to Dr. Liu's VCE Biology. Today's topic is genetic changes in a population over time, as presented in the 2025 VCE Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. When we first look at the image, we should identify the broader scientific field to activate their prior knowledge. The subject matter. The first sentence explicitly mentions genetic engineering. Students should immediately recall concepts like DNA, genes, and technological tools. The subjects. The focus is on agricultural crops, rice and maize. This tells the student that the information will likely revolve around improving a plant for human benefit. First, we identify our players, rice and maize. By including their scientific names, Orza sativa and Z maize L, the table signals that this is a formal, precise scientific study. The first story, the rice row, internal optimization. In the first row, the information column describes a process of refinement. The action, scientists are editing a specific gene called OSHXK1. The tool, they are using CRISPR-Cas9, which acts like a biological find and replace tool. The plot, this is an in-house job. No outside DNA was invited to the party. By simply tweaking the plant's existing genetic code, the narrative shift is toward energy efficiency. More chlorophyll means the plant can eat more sunlight. The second story, the maize row, cross-kingdom collaboration. In the second row, the narrative shifts from editing to importing. The action, instead of editing a corn gene, scientists inserted genes from a completely different organism, the soil bacterium Bacillus thuringiensis. The tool, this is transgenesis, moving genes across species. The plot, this is an external solution. The maize isn't trying to grow faster, it's trying to survive. By borrowing a weapon toxins from a bacterium, the maize becomes its own pesticide. Connecting, the reading between the lines. Scientists use CRISPR to edit an existing gene. Because the goal is to increase chlorophyll, the plant will likely become more efficient at absorbing light. This is a story about increasing productivity. Also, scientists took a gene from a bacterium and put it into a plant. This makes the plant transgenic. The goal here isn't to grow faster, but to defend itself. It's a story about survivability and pest control. Before looking at the crops, students must understand the Venn diagram of biotechnology. GMO, genetically modified organism. This is the broad umbrella. It describes any organism whose genome has been altered using genetic engineering technology. It doesn't matter how it was changed or where the new code came from. If you used a tool like CRISPR or a gene gun to change the DNA, it is a GMO. TGO, transgenic organism. This is a specific type of GMO. An organism is only transgenic if the modification involves the insertion of genetic material from a different species, e.g., a bacterial gene inside a plant. The rule of thumb. All TGOs are GMOs, but not all GMOs are TGOs. Row 1, rice, the internal edit. Classification, genetically modified, only. When students read the stem for rice, they see editing of the OSHXK1 gene. Because this gene belongs to the rice itself, Orza sativa, scientists are simply tweaking the plant's own instruction manual. No stranger's DNA has entered the cell. Justification. The rice is a GMO because its DNA has been altered using CRISPR-Cas9 technology to increase chlorophyll. However, it is not transgenic because no DNA from a foreign species was introduced. Row 2, maize, the biological import. Classification, both genetically modified and transgenic. In the maize field, the story changes. Scientists have inserted genes from a soil bacterium, Bacillus thuringiensis. Since a bacterium is a completely different species from maize, this is a cross-species transplant. Justification. The maize is genetically modified because its genome has been artificially changed. It is also transgenic because it contains functional genetic material sourced from a different species, a bacterium, to produce toxins. When students see a question linking genetics to yield, they should follow a three-step biological hierarchy: structure, function, result. Step 1, the structural change, the tool. The error, simply saying chlorophyll increased. Chlorophyll is a tool for capturing energy. If you have more of it, you aren't just greener, you are a better solar panel. The mark winning point: increased chlorophyll levels allow the leaves to absorb more light energy. Step 2, the functional process, that engine. The error, jumping straight to it grows more. Light energy doesn't turn into rice by magic. It must be processed through the plant's internal engine, photosynthesis. The mark winning point: this additional absorbed energy leads to an increased rate of photosynthesis. Step 3, the chemical result, the output. The error, using vague terms like better growth. Yield is measured in biomass, the grains. To make biomass, the plant needs building blocks. The mark winning point: an increased rate of photosynthesis results in a greater production of glucose, organic compounds. This extra glucose provides the energy and matter needed to develop more seeds or grains, thereby increasing crop yield. Please follow if-then formula. If chlorophyll increases, then more light energy is absorbed. If more light is absorbed, then the rate of photosynthesis increases. If photosynthesis increases, then more glucose is produced, leading to yield. In VCE, growth is a result, not an explanation. To get the marks, you must mention light energy, photosynthesis, and glucose. Without those three, you are just describing the plant, not explaining the science. Model answer: 2 marks. The increased chlorophyll levels allow the rice leaves to absorb more light energy, 1 mark. This results in an increased rate of photosynthesis, leading to a greater production of glucose, which provides the organic matter required for a higher crop yield, 1 mark. Never skip the chemistry. If the question asks about yield, your answer should almost always mention glucose. If the question mentions chlorophyll, your answer should almost always mention light absorption and the rate of photosynthesis. The Galápagos archipelago is a group of islands. The woodpecker finch, Camarhinchus pallidus, is found on 4 of the islands that comprise the Galápagos Islands. The locations of populations of the woodpecker finch on these 4 islands are shown below. This question is a subtle trap for students who rely on rote learning. The key to scoring 2 marks here is understanding the contextualized difference between comparing species versus populations. 1. The core concept, molecular homology. Molecular homology is based on the molecular clock hypothesis. Since mutations accumulate at a relatively steady rate over time, the differences in genetic sequences act as a timer for how long 2 groups have been separated. For species, we look at deep time, millions of years. For populations, we look at recent separation, thousands or tens of thousands of years. 2. The DNA record of separation. Imagine the DNA sequences of these finches are like a shared family diary. When the population on Isabella became separated from the population on San Cristobal by the sea, they stopped sharing that diary. Each island's population began to accumulate its own unique typos mutations in their DNA or protein sequences. The interpretation: if two island populations have sequences that are almost identical, it tells us they were separated very recently. They haven't had enough time to accumulate many differences. The VCE trap: students often talk about common ancestors from millions of years ago. But since these are all the same species, woodpecker finch, the narrative should focus on recent divergence and time since separation. The model answer: 2 marks. Scientists could compare DNA sequences, or amino acid sequences, between the finch populations, 1 mark. A higher degree of similarity between these sequences indicates that the populations diverged more recently, as there has been less time for mutations to accumulate between them, 1 mark. When students answer questions about molecular homology, they often use a one-size-fits-all script. However, the 2025 examiner's report for the woodpecker finch question specifically warns against this. Here is how to explain the logic behind the avoid versus use table to your students. 1. From ancient ancestors to recent splits. If you call the relationship between two groups of the same bird common ancestry, it's like saying you and your brother share a common ancestor from the Stone Age. It's technically true, but it's the wrong scale. Because these finches are the same species, their split happened yesterday in evolutionary terms. We use recent divergence point to show we understand that these populations were part of the same gene pool very recently. 2. From evolving to accumulating. Students love the word evolved, but for populations, it can be too vague or imply they've become a new species. We want to describe the mechanism. By saying they accumulated mutations since isolation, the student is explaining exactly how the DNA sequences became different. It describes a clock ticking, mutations, while the groups were apart, isolation. 3. From species to populations. This is a factual accuracy test. If the scientific name is identical, C. pallidus, they are not different species. Calling them different species is an automatic red flag for an examiner. It suggests the student didn't read the prompt. Using island populations demonstrates that the student has context to analyze the provided map and data. This is an excellent question for testing a student's ability to link a visual stimulus, the map, to the mechanical process of allopatric speciation. The examiner's comments emphasize that the marks are in the process, not just the name of the phenomenon. 1. The barrier, isolation. The story starts with the map. Notice the distance between San Cristobal and the other islands. The finches on San Cristobal are physically separated from the others by the sea. Because they cannot fly that distance, they are geographically isolated. This means the phone lines are cut. There is no gene flow between San Cristobal and the other populations. 2. The change, selection and mutation. Once isolated, the finches aren't just sitting still, they are responding to their specific environment. San Cristobal might have different food sources or a different climate. Consequently, the population experiences different selection pressures. Meanwhile, unique mutations are ticking away in their DNA, distinct from those on the other islands. 3. The advantage, survival. This is where students often lose the mark. You must mention how they change. Birds with traits that are advantageous for San Cristobal, such as a specific beak shape for the local insects, have a selective advantage. They survive, reproduce, and pass those specific alleles to the next generation. 4. The final split, speciation. Eventually, the two groups become so different that if they were reunited, they could no longer interbreed to produce fertile offspring. The model answer, 3 marks. To get full marks, the student must refer back to the map. The woodpecker finches on San Cristobal are geographically isolated from other populations by the sea, preventing gene flow, 1 mark. Because San Cristobal has different selection pressures, individuals with advantageous traits of alleles have a selective advantage, and are more likely to survive and reproduce, 1 mark. Over time, different mutations accumulate in the isolated population until they are genetically distinct and can no longer interbreed, a process known as allopatric speciation, 1 mark. Avoiding the sympatric trap. The examiner's report was very clear: this is not sympatric speciation. Why? Sympatric speciation happens in the same location, like different breeding seasons in the same forest. The map proof. The map clearly shows the populations are in different locations. If a student writes sympatric, they have ignored the most important piece of evidence provided, the C. Sympatric speciation, the invisible wall pitfall. The concept, speciation, occurs while the populations are still living in the same area. Common pitfall, students assume that if they live together, they must interbreed. They miss the invisible, reproductive barriers. This is like 2 groups of people living in the same city, but speaking different languages and never meeting. One group might only come out at night, temporal isolation, or they might only respond to a specific type of music, a behavioral isolation. Key requirement: you must identify a non-geographic factor, such as polyploidy, extra chromosome sets, mating seasons, or mating calls. Allopatric speciation, the physical wall pitfall. The concept, speciation, occurs because a population is split by a physical geographic barrier. Common pitfall, students often forget to mention gene flow. They say they were separated, but they don't explain that the separation stopped the exchange of alleles. Think of the Galápagos map. The sea is the wall. Because the finches on San Cristobal cannot reach the other islands, their gene pool is locked in a room by itself. Key requirement: you must state that there is no gene flow due to geographic isolation. Exam strategy: the one-question test. Teach your students to ask themselves this one question when they see an exam prompt. Are they physically touching, or could they walk or fly to each other? If no, it is allopatric. Focus on the physical barrier. If yes, it is sympatric. Focus on the timing or behavioral change. The allopatric logic chain for full marks, GSA. Isolation: identify the geographic barrier, e.g., the sea. Gene flow: state that gene flow has stopped. Selection: mention different selection pressures or mutations. Advantage: mention that individuals with selective advantages survive and reproduce. Question 4. Genetic engineering in crops. All TGOs are GMOs, but not all GMOs are TGOs. Rice, CRISPR editing of OSHXK1 equals GMO only, maize, BT gene insertion equals GMO plus transgenic. Yield chain, chlorophyll light absorption photosynthesis glucose crop yield. Question 9. Speciation in Galápagos finches. Molecular homology. Compare DNA amino acid sequences to determine relatedness. Focus on recent divergence, not common ancestry, when comparing populations. Allopatric speciation. GSA geographic isolation, different selection pressures, advantageous allele speciation. Key exam tips. Always mention gene flow when discussing speciation barriers. Use precise vocabulary: populations, not species; accumulated mutations, not evolved. For yield questions, always include light energy, photosynthesis, and glucose. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.